And our speaker is well known to you, is Dr. Altaf Kondakar, which he is an associate professor and head ophthalmic genetics laboratory in the Department of Ophthalmology, uh, University of King Saud University. And he received his PhD degree in applied biology from University of Mumbai in India, and he has done his postdoctoral fellowship uh, from Baylor University in College of Medicine, Houston, Texas, United States of America, and Thomas Jefferson University, Philadelphia, again USA. Before he joined the department, he has a research interest in the area of genetics and genomics of clinical transitional research in complex human diseases. He's been working with us over the last 10 years in the Department of Ophthalmology as a head of the genetics laboratory in our department. His current research focuses on identifying genetic and epigenetic components involved in the ocular diseases with a major interest or focus on glaucoma. He has contributed to the literature around 70 publication. What he will do today, he will update us on the genes and genomics, on genetic loci associated with primary angular closure glaucoma and their principal mechanisms by which these gene and loci may increase the risk of the disease and contribute to the primary angular closure pathogenesis, angular closure glaucoma pathogenesis. Dr. Altaf, please welcome.
Good afternoon, everyone. Thank you, Professor Ali, for the kind introduction and your time for moderating this session. Am I audible? Can I be heard clearly? Can you hear me clearly?
Hello?
Can you hear me?
Yes, I can hear you, but I don't know about the audience.
Okay.
Okay.
So, thank you very much. So, as Professor Ali pointed out, I will. I'm trying to share my screen right now. Please give me a moment.
Yes, it's clear. Yeah. Okay, good.
Is the screen clear now?
Yeah.
Setting.
Is it clear for the audience everywhere or the who are joining us? I hope it is clear, the slides.
I think so.
Yeah, go ahead.
Okay. Thank you again, everyone. Very good afternoon to all of you. Thank you, Professor Saleh, for your kind words and your time to moderate this session. As Professor Saleh pointed out, I will talk today about the genes and genetic mechanisms that are associated with primary angle closure glaucoma. And before that, just briefly, I will touch upon some basic clinical and epidemiological aspects of PACG. So, what is glaucoma? Glaucoma is classically defined as a group of optic neuropathies that are associated with progressive loss of retinal ganglion cells, which lead to visual field loss, appearance of characteristic cupping, and degeneration of optic nerve head, which eventually leads to blindness. This is an example of how glaucomatous cupping looks like to your right, and it's compared to the normal optic nerve head to the left. So why is glaucoma important? So why is glaucoma important? So this is a pie chart which represents the global causes of blindness that is seen. And as you can see, um, according to World Health Organization, glaucoma is the second leading cause of blindness just after cataract. Now, this is an actually old data of from 2013, but I'm sure even the recent data, the distribution will be more or less similar. And besides blindness, glaucoma affects the quality of life. It is also a socioeconomic health burden, and more importantly, it can be inherited. And furthermore, important is it can be prevented and managed by treatment if diagnosed early. So, hence the need to understand the. Sorry, okay. So, where we were, I was actually trying to say that glaucoma is a very common cause of blindness, and besides the blindness, it can affect quality of life. It's a socioeconomic health burden, and it can be inherited. Plus, it can be prevented and managed by treatment. So, hence, there is a need to understand the mechanisms and there is a need for early diagnosis. So, what is the known pathogenesis of glaucoma? Glaucoma is usually, but not always, associated with IOP elevation, and IOP elevation is generally due to increased aqueous outflow resistance. So this is a simple diagram which shows how the aqueous outflow pathway looks like. Now, the things in the blue arrow shows actually the flow of aqueous humor that is produced by ciliary body that passes from the posterior to the anterior side via the conventional trabecular meshwork into the skin canal. And this is the pathway which is more very conventional and is dependent on IOP. And besides the trabecular meshwork, there is another pathway which also exists where a small portion of aqueous humor flows through which is called as a US scleral outflow pathway. And this is actually a pathway which is IOP independent. Now, what happens in primary open angle glaucoma is there is increased resistance to the aqueous outflow through the trabecular meshwork, as it is seen in this figure. And as compared in contrast to in angle closure, there is an occlusion of the TM by the peripheral iris. As you can see over here, the flow is interrupted, which leads to, so there is occlusion of the TM by the peripheral iris, which leads to obstruction of the aqueous flow. So what are the mechanisms of angle closure, the clinical mechanisms which include? So there are two types of forces. It is hypothesized that there are two types of forces involved: the forces that push the iris forward and the forces that pull the iris against the trabecular meshwork. And classically, they are divided into two types, which are pupillary block and the non-pupillary block. The pupillary block is mainly due to the proximity between the lens and the posterior face of the iris that actually forces the iris to bow anteriorly. And in contrast, in the non-pupillary block, there are other factors like the thickness of the peripheral iris, the anterior location of the peripheral iris, the anteriorly rotated ciliary body, and plateau iris. These are some of the UBM images of the types of narrow angles. I have taken it from this paper in the Progress in Retinal Eye Research. So here they reflect the relative pupillary block, example of a plateau iris, a thick iris with plateau iris, and there is a ciliary choroid detachment with complete closed angle after an acute attack. This is an example of anteriorly rotated ciliary body with closed angle and small lens to ciliary body distance. And this is an example of ciliary body cyst with narrow angles. So, and accordingly, these angle closures are classified as primary angle closure suspects, where there is a narrow angle is suspected, and but these people have normal IOP with normal optic disc and visual field. And the second one is where the actual angle closure has taken place, and such patients they normally exhibit a high raised IOP. And the third is with where actually the glaucoma sets in, where they have features of primary angle closure combined with evidence of glaucomatous optic neuropathy. Now, this is just a brief about the epidemiology of primary angle closure glaucoma. Recent systemic reviews and meta-analysis have shown there is increase in prevalence of glaucoma. As you can see here, the worldwide population of glaucoma is expected to reach 111 millions by 2040, and that of angle closure glaucoma is expected to exceed 30 million by 2040. Besides this, the prevalence of PACG also greatly varies with geographical regions and ethnic groups. So this is a bar chart which shows that how the prevalence varies. You can see there is the incidence is different among different population. When it is very highest in among Asia and including Middle East. Middle East also has very high prevalence of PACG as compared to the European counterparts, indicating that there is racial differences. Now, this is a pie chart which shows the glaucoma pattern reported at KUH. This was done at our hospital and by Professor Vedan and groups, which was published in 2011. So this is again an old data, but here it can be clearly seen that PACG was the predominant form of glaucoma, which was followed by PAC and then angle closure glaucoma. Of course, this does not reflect that the PACG and prevalence is very high, but at least it supports the finding that, as you saw in the previous slide, that the PACG is far common in the kingdom and in Middle East. Besides that, PACG is also more common among women. It is reported that 60% of women are susceptible to PACG. And as compared to POAG, in the general population in the age group of 40 to 80 years, the prevalence of PACG is lower as compared to POAG, but the likelihood of severe bilateral visual impairment in PACG is 3 times higher as compared to POAG, thus posing a significant public health concern. So what are the risk factors that are involved in primary angle closure glaucoma? As we saw in the previous slide, there was mention of gender, where we saw it was more common in women. It is as age increases, glaucoma itself is a disease of the aging. So age is also a risk factor. And plus, in angle closure glaucoma, there is more contribution of anatomical risk factors, like small cornea, small anterior chamber, axial length, and differences in the iris, the location of the iris, and the lens. Plus, genetics also contributes towards the angle closure, and there are certain reports where even environmental factors have been known to influence the occurrence of angle closure glaucoma. So now we'll look at the genetic aspect of angle closure glaucoma. So all these things point out that this disease is very multifactorial with a complex etiology. So let's see what are the PACG and genetic links. So coming to the genetic link, first degree, there are studies have reported that the first degree relatives of of subjects with PACG, they have 3 to 5 times greater risk of developing the disease. Besides, the heritability of narrow angles was almost 60% in the first degree relatives of patients with PACG, uh, 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 PAC and PACG. The risk of narrow angles was 7.6 times, and that of angle closure was 13.6 times higher in siblings of the affected patients. So you can see the risk is very high in the first degree relatives as well as in the relatives, in the siblings. So due to high heritability and increased risk among the family members, particularly siblings, are consistent with the observation that genetic factors do play an important role in narrow angles as well as in primary angle closure glaucoma. In addition to this, the initial mapping of nano-ophthalmus and NO1 gene in large family of patients with autosomal dominant nano-ophthalmus, hyperopia, and severe late-stage phenotype of angle closure was the first strong evidence for a causal involvement of the genetic component of in PACG, where they are shown that a gene which is involved in ocular development was involved in PACG. And since then, several studies have been investigated to find for associations and genes that are involved in PACG using different approaches. So what are these different approaches that have been used to identify the genes in primary angle closure glaucoma? So there are a number of approaches that have been used that include genome-wide analysis, candidate gene analysis, analysis using quantitative trait loci. Animal studies have been used to identify these. There have been expression studies that have been performed, and linkage and even the new generations, next generation sequencing have also been recently used to identify genes in PACG. So we'll have a look one by one of what all these approaches have led to the identification of which genes. Coming to the first one, which is the genome-wide association study, which is commonly referred to as GWAS. So this is basically a hypothesis-free approach, where the study looks for association between genetic variations which are spread across the entire genome with a particular disease in a large population of cases and controls. The analysis actually gives highlights polymorphisms which are associated with the disease, and these polymorphisms can be present inside the gene or between the gene. So now these genes then serve as a candidate genes. Further analysis of these genes with respect to their known functions and pathways that are involved in gives the insight into the mechanisms, and they serve as a basis for future studies. So this is basically just shows the what GWAS normally overall view of GWAS. So now in angle closure glaucoma, there are two major GWASs that have been performed. Um, and these GWASs involved a discovery stage where they identified genes, and then the replication stage. So coming to the first GWAS, as I mentioned that this involved a discovery stage and a replication stage. So in the discovery stage, they utilized almost more than 12,000 subjects, samples across five independent collections in Asia, which included Singapore, Hong Kong, India, Malaysia, and Vietnam. And once these genes were identified, they were subjected to replication. In a replication stage, which again involved equal number of samples, more than 10,000 subjects were there, samples were there, across six independent collection sites. That two were from China, one from Singapore, India, Saudi Arabia. Our lab was also participate, which are part of this collaboration in the replication study, and also from UK. So these are the results that were observed in the GWAS one. This is a representation of the Manhattan plot. It is called. You can see on the x-axis, is the chromosomes starting from 1 to 22. The y-axis represents the p-values, and each spot what you see over here represents actually a polymorphism, or SNP as it's called as. And this cluster of colors, what you can see here, actually they are the cluster of these spots which are non-significant. And this bar, what you can see line, is the genome-wide significance line. Any SNP, any dot or SNP which is above this line are referred to as significant SNPs. So here, this GWAS one showed like these were the three SNPs that were found to be highly significant in both the discovery stage and the replication stage. And these two polymorphisms were actually found to be localized to call 11A1 and FLAC-A7 as candidate genes. Whereas the third polymorphism was found to be located in the intergenic region between PCN-PD1 and ST18 gene. So this is the all the results of the GWAS one. We can see three variants were identified, and they were localized to these three genes, which were both in the stage 1 and stage 2. They were found to be highly significant. These were the risk allele, and these are the odds ratio of these polymorphisms that were found to be associated with. Now we look at one by one what is the possible role of these genes in PSCG. Coming to the first one, which is called 11A1, this encodes alpha chains of type 11 collagen, which is a relatively minor fibrillary collagen. And mutations in this gene have been previously associated with type 2 Stickler syndrome and Marshall syndrome. And these syndromes are associated with manifestations of high myopia and blindness from retinal detachment. This is actually an mRNA expression of this called 11A1 in different types of ocular tissues. This is an endogenous beta actin gene which is used as a control to keep a check on the false negatives, and this NC refers to as a negative control which keeps a check on the false positives. So these are the different types of ocular tissue samples, right from cornea to trabecular meshwork to iris, lens, and it can be clearly seen that COL11A1 is expressed in most of the ocular tissues. So what was the hypothesis for possible role of COL11 in PSCG? Eyes predisposed to PSCG are generally small and hyperopic with shorter axial length. So this variant was hypothesized to alter the function of this gene in such a way to persuade refractive area development that may result in small hyperopic eyes and thereby predisposing to PSCG. Besides, abnormality of collagen in scleral and lamellar fibrosa may also increase susceptibility to axial length changes and may predispose to PSCG. And also there was a third hypothesis considering that the presence of COL11 in human TM cells, it is also possible that the differential expression of this gene in the TM may have a critical role in regulating the aqueous outflow pathway to influence the disease risk. Now coming to the second variant, which was found to be located in the intergenic region between PCM-TD1 and ST18. PCMTD1 encodes a protein called isoaspartate O-methyltransferase domain. This basically there is no known function of it yet. Whereas the ST18, which is known as the suppression of tumorigenicity 18, encodes a zinc finger binding DNA transcription factor, which is known to be tumor suppressor in breast cancer, and it's also a regulator of pro-apoptosis and pro-inflammatory genes in fibroblast. Like call 11A1, this gene was also found to be located in major ocular tissues. And interestingly, it was observed that ST18 was not expressed in TM and the iris, suggesting that possibly among these two, PECM-PD1 would be a more likely candidate for angle closure glaucoma. So what is the possible role of these two genes? As I mentioned, that these both of these genes role is not clearly known right now in PSCG, but there has been studies reported where this variant was found to be associated with PACS, which is the angle closure suspects in Indians and Chinese patients. And as I mentioned earlier, that PACS is the earliest stage of PACG, suggesting that this locus might increase the risk of narrow angle configuration. Coming to the third locus, which was identified as FLAG-A7, this is the gene which encodes a protein called plextin homology domain containing protein 7, which is an adenine junction protein that plays a significant role in maintaining the stability of adenine junctions to regulate the paracellular permeability and cell-to-cell adhesion. And there has been recent studies to better understand the possible role of molecular and cellular contribution of this gene to PACG pathogenesis. This gene is also found to be expressed in all the ocular tissue. This is a bar chart which is compared to the endogenous as compared to the beta-actin endogenous control, and you can clearly see this gene is also expressed in all the major ocular tissues, supporting its role in PACG. This gene has been hypothesized to influence uh, fluid dynamics within the eye. The localization of like A7 protein in the vascular endothelium, particularly within the microvasculature of the iris, ciliary body, and choroid, raises the possibility that like A7 would be playing an important role in regulation of vascular permeability and intracellular fluid dynamics within the eye. Changes in the iris volume during pupillary dilation and choroidal effusion have been suggested to be involved in the pathogenesis of PSCG. Besides, this gene has been reported to be downregulated in lens and iris of PSCG patients, which were found to also be correlated with the risk of this variant, risk allele of this variant, emphasizing their role in PSCG. Besides the maintaining the fluid dynamics, this gene has also been hypothesized to maintain the blood aqueous barrier permeability. Confocal microscopy experiments have demonstrated the presence of this gene at adherent junctions and tight junctions of the apical junction complexes that play an important role in maintaining blood aqueous barrier stability. And silencing of this gene in primary human TM cells and immortalized NPCE cells, which these two are key components of the aqueous flow pathway and the blood aqueous barrier and play an important role in glaucoma. Silencing of these genes was found to affect actin cytoskeleton structure, cell migration, and permeability via the GTPase signaling pathway involving RAC1 and CDC42, suggesting that the barrier defects due to reduced FLAG-A7 level contributes to the etiology of PSCG. Disruption of blood aqueous barrier and leakage of inflammatory proteins and cells into the anterior chamber of the eye, as is observed in the acute and chronic angle closure glaucoma, has been suggested to contribute to increased IOP and could also be one of the mechanisms in angle closure. So what is the possible role of FLAG7 in PSCG? It has been hypothesized that this variant in this gene may lead to reduced expression of FLAG7, leading to leaky blood aqueous barrier due to reduced tight junctions and adherent junctions proteins and altered regulation of fluid dynamics across the squamous canal, which will result in elevated IOP and clinical manifestations of PACG. However, these hypotheses have not been validated using in vivo functional studies and animal studies. So to summarize the findings of GWAS1, Three genes were identified, and three variants were identified in PSEG. As you can see here, I have reported some more variants. This is which I'm not talking about in this talk, but there are some more variants that were reported in collagen A1, which were also found to be associated with PSEG, as well as severity of the disease and in angle closure. I'm sorry, anterior chamber depth levels. So these are the different mechanisms that we just discussed, how these three genes can have their effect on via acting via the cytoskeletal organization or by ECM organization. The functions of PCTMD is still unknown, whereas ST18 can affect through apoptosis or inflammation. Now coming to the findings of GWAS2. So GWAS1 was expanded to over 40,000 participants, including 10,000, more than 10,000 PSEG cases across 24 countries. And again, our lab also participated, was a part of this collaboration in the replication cohort. This study has also been published in 2016 in Nature Genetics. Possibly this is one of the largest GWAS that has been performed so far in any glaucoma types. And this study reported five new loci in addition to the what previously three loci that were reported. This is again a Manhattan plot of the GWAS2 findings. As you can see here, the GWAS1 results were replicated. Whatever the genes that are underlined, they again showed up in this study as well. Besides this, they identified five new loci, which are included EPDR1, GLIS3, DPM2, FAM012, CHAT, and FAM2, which have been circled in this slide over here. There was another gene, as you can see over, which is FNDC3B, which showed up in the discovery phase, but was not validated in the replication cohort. So this is, I've just tabulated all those, the list of the variants that were identified and the gene loci where they were found to be located. All these genes were found to be significant more at the monomoid level, and this is the odds ratio of per allele. So coming to each one of these genes, now we'll see how these genes play a role in PSEG. First is the FAMT1, which is Formitin family member 2. It is also known as King2 and MIG2, which is a Kindlin2 hematogene inducible gene. This gene FAMT2 encodes a plextin homology domain containing family C1 member 1 that belongs to the same plextin family of protein as we saw earlier slide of flex 7. This protein is a component of extracellular matrix and plays a role in integrin activation and cell-to-cell adhesion and actin cytoskeleton assembly. All these processes play a significant role in development and progression of glaucoma, thereby implicating the role of MT21 in PACG pathogenesis. There is no further studies on characterization of this gene yet in PACG. Coming to the second one, which is EPDR1, this is a lysosomal protein of unknown function, and it encodes a glycosylated type 2 transmembrane protein related to the families of cell adhesion molecules and may have a similar role in PACG. A recent crystal structure study revealed that EPDR1 functions as a lysosomal activator protein or a lipid transporter. Previous genes that were identified in POAG had identified proteins like MyoC, caveolin-1, and APCA1 that played a similar role of removal or repair of damaged lipids and influencing the membrane surface tension in glaucoma patients. Considering that role, the role of EPDR1 in as a lipid transporter, it is postulated that it might have a similar role in PSAG, but it needs further study investigations to support this hypothesis. The third candidate, which was GLIS3, is involved in it's a transcription factor which is involved in multiple cellular processes. Interestingly, this gene has been identified as susceptibility locus in diabetes. In fact, knockdown of GLIS3 has shown to increase beta cell apoptosis induced by pro-inflammatory cytokines such as interleukin beta 1 and interferon in rat models. Likewise, GLIS3 upregulation was found to protect against cytokine-induced apoptosis. The GLIS3 also functions as a Wnt activator and as activator of its Wnt signaling pathway. And GLIS3 expression is hypothesized to affect the development of cell survival of the tissues of anterior segment structures and thus influence the PACG risk. The fourth loci, which was found to be located in between DPM2 and FAM0, 1, and 2, these are uncharacterized genes yet. And DPM2 encodes a protein which is involved in glycosylation and has been associated with congenital disorders of glycosylation which were related to nervous system. Altered glycosylation has been reported in glucomatous TN, suggesting that these alterations may elicit biological and biochemical changes in the ECM, which in turn can contribute to the glaucoma pathophysiology. And the second loci of FAM102A was earlier identified as an estrogen-induced gene 1, EEIG1, in response to 17-beta estradiol, suggesting a role in estrogen metabolism. As I mentioned earlier, that PACG is far more common among women as well. So this gene might have a gender and hormone might influence the glaucoma outcome with using this pathway. Besides, FAM102A has also been shown to as a novel receptor activator of NF-kappa B signaling with a role in osteoclast formation, suggesting its role in tissue maintenance, repair, and remodeling. The fifth loci that was identified, which is CHAT, cholinor-acetyltransferase, is an enzyme which is involved in the synthesis of neurotransmitter acetylcholine, which has a role in pupillary constriction. Interestingly, anticholinergic agents have been found to mediate pupillary block and increase acute PSCG risk. RGCs have been also shown to possess a viable chat system which may help regulate acetylcholine synthesis and function. And considering the potential role of cholinergic system of the eye, it is possible that genetic variants in chat gene could alter the risk of PSCG by regulating acetylcholine metabolism and influence the autonomic regulation of PSCG related structures. This is again a combined slide of showing expression of all these genes that were identified in the second GWAS present in most of the ocular structures. mRNA expression of EPDR1, class 3 from to DPM2 and was observed in all the ocular anterior segment tissues involved in iris, ciliary body, and the TM. So to summarize the findings of GWAS2, they identified 5 new loci as well as they confirmed the previous genes that were identified, pure polymorphisms that were identified in PSCG. And these are the possible functions of all these 5 genes. From to which I just discussed, they were found to play a role in ECM addition and signaling. EPDR1 can serve as a lipid transporter. GLIS3 can have a role as cell survivor or a activator. DPM2 can have a role in glycosylation. FAM2 may have a role in estrogen metabolism and hence RANK signaling, whereas CHAT can play a role in acetylcholine metabolism and control autonomic inhibitions. Now coming to the second approach, which has been used to identify genes in PSCG, is the candidate gene analysis. Unlike genome-wide approach, the candidate gene approach focuses on identifying association between genetic variations in a pre-specified gene of interest and a pre-specified polymorphism in cases and controls. Several studies have examined the association between genetic variants in at least 5 different genes in PSCG. And recent meta-analysis have revealed 5 candidate genes that may contribute to a PSCG risk. And these include matrix metanoprotease 9, which is MMP9, nitric oxide synthase 3, heat shock protein 17, membrane type fusion-related protein, MFRP, and hepatocyte growth factor, which is HGF. We'll see one by one what these genes, how they play a role in PSCG. So in MMP9, there are a number of polymorphisms that have been reported that are found to be associated with PSCG. And this gene encodes gelatinase or type 5 collagenase, which is actively involved in ECM remodeling, RGC cell death, and represents an important candidate. In fact, MMP null mice have been shown to have aberrant collagen composition in the TM, which causes reduced aqueous humor drainage and elevated IOP. MMP9 was found to be needed to enhance outflow and maintain IOP homeostasis. So that's the known mechanism in PSCG. And these gene polymorphisms have been associated with a number of ethnic populations like Taiwanese, Han Chinese, Australian, Indian, and Pakistani population. The second gene, which is the NOS3, again, there are multiple polymorphisms in this gene that have been associated with both angle closure glaucoma and anterior chamber depth. This gene, as we all know, is involved with the formation of nitric oxide, which is an important biological mediator in neurotransmission, vascular tone regulation, vasodilatation, and apoptosis. It has been implicated in several neurodegenerative disorders, including glaucoma. NOS expression and NO production has been shown to enhance MMP9 activity, suggesting that the nitric oxide and MMP9 related pathways may have an important role in PACD pathogenesis. However, unlike in POAG, where the relationship between vascular dysregulation has been clearly defined, as, and the vascular dysregulation in PACG is not yet clearly identified. The third candidate gene, which is the heat shock protein 78, which is also known as HSPA1A, a polymorphism has been reported that is associated with PACG. This gene encodes a stress response protein that functions as a molecular chaperone to regulate protein folding, translocation, misfolding, degradation to maintain protein homeostasis and cell survival. Variation is that this gene is located in the 5' untranslated region and is known to downregulate the expression of heat shock protein 17. HSP17 has also been implicated in RGC cell survival, and like NOS3, HSPA1 also induces transcription of MMP9 through activation of NF-kappa B and activating protein 1, and may have an indirect role in PACG. The fourth gene, which is the MFRP, which is a membrane phaser related protein. Again, number of polymorphisms have been reported in PACG where this gene encodes a frizzle related protein family, which is involved in prenatal and postnatal ocular growth and development. This gene has already been involved in axial length regulation, known to cause nanophthalmos, posterior microphthalmia, and retinitis pigmentosa. And based on its structure, which is a frizzle-related protein, it is known to be regulator of light signaling pathway. It has been hypothesized that the variant in this gene may lead to abnormal changes of ocular structures in early age, conferring predisposition to angle closure glaucoma. The fifth candidate gene, which is a GF, which encodes a hepatocyte growth factor, is a secreted mesenchymal cell, is an active polypeptide, which when activated binds to mesenchymal epithelial transition factor to activate HGF-c-MAT signaling and stimulate cell growth, migration, morphogenesis, and angiogenesis in numerous tissues and cell types. And overactivation of this signaling pathway has been found to promote cancer development and can also stimulate various downstream signaling pathways, including Wnt and beta catenin signaling, which they all play an important role in glaucoma. In fact, this polymorphism in HGF have been associated with refractive errors and keratoconus. And GF receptors are expressed in TM and overexpressed in glaucomatous eyes. They are known to confer protection to RGCs by increasing neuronal survival and promoting hexagonal regeneration. Given its role in ocular tissue, it is speculated that these variants may affect HGF expression and overactivate GF-CMAT signaling pathway to induce morphological and physiological changes of anterior segment structure, thereby disrupting normal aqueous regulation and increase the risk of PACG. Now, besides these five candidate genes, based on the hypothesis that some genes reported in other form types of glaucoma, such as POSAG, POAG, or congenital glaucoma, these genes were also examined in PACG, like myosin, and optimurin, or WTR1 and CYP1B1. They were investigated in PACG as well, and they were not found to be associated in our population. However, there have been reports of myosin involved in PACG from Quebec, whereas CYP1B1 haplotypes were found to be associated with PACG subjects of Indian origin. In addition, genes in antioxidant system and oxidative stress pathways have also been evaluated in PACG, such as the superoxide dismutase 2 catalase, glutathione S transferase, and mitochondrial DNA. And these are the polymorphisms that have been studied. However, these results indicated that the minimal role of oxidative stress in angle closure glaucoma as opposed to open angle and pseudo exfoliation glaucoma. And recently, we have investigated, we reported a gene involved in BMP signaling pathway, which is the ACVR1. This, for the first time, we reported a genetic variant, RS1297, in the 3' UTR region of the gene that was found to be associated with PACG. This ACVR1 is a member of BMP signaling pathway, which is a bone morphogenic protein type 1 receptor that belongs to the TGF beta family, and they are involved in BMP signaling. In fact, members of this BMP family of genes have been known to express in the human trabecular meshwork and optic nerve head. So, what is the possible mechanism by ACVR this variant can cause be involved with PACG? ACVR1 is reported to be a Wnt signaling antagonist. loss of ACVR1 was found to increase osteogenesis by activating Wnt signaling and decreasing the expression of Wnt inhibitors. So we hypothesized that this variant, since it's located in the 3' UTR region of the gene, may affect the binding of microRNA, particularly 333P, and result in overexpression of the ACVR1 and negatively regulate, which might in turn negatively regulate Wnt signaling via these Wnt inhibitors, and which will inhibit the Wnt that may downstream affect ECM, TM homeostasis and causing ECM abnormalities. Besides this, there have been other recent study which has reported SMARC2, which is a spark-related modeling calcium binding protein. A polymorphism has been reported in the Saudi population from Riyadh Hospital, I think this was, where they reported a variant which encodes a metrocellular glycoprotein and is known to regulate expression of ECM proteins and metanoproteinases, which was associated with the PACG patients of Saudi origin. Now coming to the third approach, which is identification of genes using quantitative traits. Now, what is basically what is this all about? So, genetic dissection of complex multifactor disease can be done by identifying direct association of genes with the disease phenotype, as we saw right now, where they were in GWAS or using candidate gene approach. They use these approaches to have a direct association of genes with the disease phenotypes. However, you can also use an intermediate phenotype to find what are the genes involved in this, and then you can associate it to the disease phenotype. So what are these endophenotypes? In PACG also studies in the past have demonstrated that ocular biometric parameters such as shallow anterior chamber depth and short axial length are strong anatomical risk factors of PACG, among other ones related to iris and the lens. So a GWAS was performed and they found that this gene called a variant mapped to ABCC5, which is an ATP binding cassette subfamily C member 5, was found to be significantly associated with shallower ACD in a large data set of case control studies from Asia. Again, this was a multi-ethnic group, and we were also part of this, our lab was also part of this study. So they identified this gene, which is also expressed in anterior segment eye structures such as iris, ciliary body, lens, and cornea. Although the exact mechanism of this gene in angle closure glaucoma is not known, however, the inhibition of endogenous ABCC5 in zebrafish was found to show a significant reduction of body length and ocular size, suggesting a role of ABCC5 in eye growth and development. Coming to what animal studies have been used in to identify these genes in PACG. Now, there is a gene called serine protease 56, which was a mutation was identified in a seri- in sorry, a mutation in serine protease 56 gene was identified to be associated with decreased AL in a mouse model that was resembling acute closure angle closure glaucoma-like phenotype. Besides, mutations in human CREST6 were also found in 6 families with recessive posterior microphthalmia and were also found to be significant, which caused a significant reduction in ocular length in the same study. So this gene was identified in the mouse as well as in the human subjects, which were associated with shorter axial length. In fact, recently the same group has showed that the loss of CRIS56 function leads to angular angle defect and high IOP in PCR 56 deficient mice. They suggested a critical role of this gene in development and maintenance of ocular drainage tissue and IOP homeostasis. There is another gene called calcitonin receptor-like receptor, which is calciral, which was identified in transgenic mouse model of acute PAC. Overexpression of these genes was found to result in pupillary palsy and angle closure due to relaxant effect of adrenaline in the pupillary sphincter muscle, leading to obstruction of aqueous outflow and acutely and transiently elevated IOP. So this gene was overexpression of this gene was found to lead to acute PAC in transgenic mouse. The calciral belongs to a family of G protein coupled receptor and functions as a adrenaline receptor. It was hypothesized that defective adrenaline receptor regulation in pupillary sphincter muscle might result in the development of acute attack of angle closure in humans. In fact, there have been two studies in the human subjects of PACG where this gene polymorphism has been associated with both with acute angle closure as well as chronic angle closure in Chinese as well as I think in Australian cohort. So what are the gene expression studies that have been used in PACG now? So there are not much, but there is one study that has reported increased expression of SPARC, which is a secreted protein acidic and rich in cysteine protein, gene, which was found to be increased. This spark, which is spark as well as collagen 1 transcript were reported to be elevated in PACG iris. It was proposed that spark through its effect on iris ECM composition is likely to contribute to changes in the iris biomechanics that might lead to angle closure glaucoma. In fact, spark null mice have been shown to have lower IOP with a greater turnover of aqueous humor outflow, suggesting that spark is involved in outflow resistance. However, as I said, there are no systematic studies of blood-based or tissue-based actual mRNA profiling in PSAG. So this was only one study that was reported overexpression of mRNA. Now, as we saw this so far, that candidate genes and GWAS genes have been used to identify genes in PSAG. And now recently, whole exome sequencing, next generation sequencing technologies are emerging, and they have been also being used in PSAG. So these are some recent publications that have identified genes using exome sequencing. Using a linkage as well as a whole exome sequencing, these group identified a genetic cause of corneal angle closure in three unrelated Iranian families. This is a call 18-1 gene, which was mutation in this gene were identified. This gene is expressed in UNT and cornea and ciliary body. And type 18 collagen is a component of ECM matrix and is also involved in vein signaling. This is a second study that is recently reported that they found a mutation using the same approach of linkage as well as whole exome sequencing. They identified a causal variant in spermatogenesis associated protein 13, which is Sparta 13, in a large PSAG family of British origin. Sparta 13 is suggested to have a role in cell division and cell adhesion in the anterior segment of the eye, thereby affect tissue homeostasis and influence PSAG pathogenesis. So, so far, whatever these genes that have been identified, if you do a simple pathway enrichment analysis of all these genes that I mentioned about, it indicates. I'm sorry for this. This is a very busy slide. This is our interaction of all these genes that I mentioned about, and importantly, what it shows is this big large block, which is a pathway involved in extracellular matrix organization, which indicates actually that ECM remodeling may be a key mechanism in angle closure glaucoma. So to summarize, so far, GWAS investigations have revealed 8 genetic loci in FLEC-A1, COL-A1, PCTM-U18, FUN2, ETP, EPDR1, CLAS3, and CHAT that may have significant role in PACG development. Additionally, candidate genes have identified another 5 genes that have been suggested to influence the risk of PACG. Besides, there have been certain important candidate genes have been reported recently, and the animal studies and expression studies genes have also shown to be promising. The GWAS quantitative traits have identified a gene called ABCC5, which have a role in anterior chamber depth. However, in the absence of any direct functional evidence, the mere presence, as I mentioned, these genes are known to be present in most of the ocular tissue. However, the mere presence does not confirm their role in in PACG etiology, and therefore further in vivo functional and molecular and animal model studies are needed to actually elucidate their causal role in PACG pathogenesis. And as has been observed in complex human diseases, this loci account for relatively small fractions of PACG, less than 5% or maybe, with moderate effects indicating the presence of additional genes or genetic variants that may have been missed or not covered, like example, the rare variants, insertions or deletions, or copy number variants that remain to be explored. Also, additional roles of gene-to-gene interaction, transcription signatures like mRNA and non-coding RNAs or epigenetic regulators also remain to be investigated. Next generation sequencing, as you saw, there are 2 studies that reported identification of the 2 genes in PSAG also represents an important tool in PSAG. But I think that will be more important to inhibit families that show strong familial links or to identify rare variants. So what I feel is that using integrated genome analysis consisting of whole genome or exome sequencing combined with transcription analysis or epigenetic regulation might highlight the underlying genetic and environmental abnormalities that may be important to PSCG clinically. Until then, the importance of accurate clinical phenotyping cannot be overemphasized, and the diagnosis of angle closure between iris and trabecular meshwork remains the hallmark of PSCG. Although to date the translation of genetic findings to clinical practice in PSCG is limited, however, genetic research continues to unfold an understanding of the molecular pathophysiology of PSCG. And hopefully in the future, it may be possible to development an algorithm of possible polygenic risk scores to assess future risk of PSCG in patients and make it clinically useful. These are some of our studies that are published in PSAGs, which is already published. These two were published in Nature Genetics, and then the ABCC5, as I mentioned, were part of the replication cohort, and there are some other gene mutations and the one which we recently published. I would like to acknowledge all the members of our research team who contributed towards the work that I used in this talk, from a clinical side, from our research unit and the support staff, and some of our past investigators, our collaborators, and the Glaucoma Research Chair for their continued support. I acknowledge their support in this. And thank you very much for your attention.